Okay, so this is going to be chapter 17, okay? So we're going to be talking about DNA. Okay, so a couple things that we need to know is it goes DNA to RNA to the protein. Okay, and the proteins are made up of amino acids that are linked together by amide bonds. Okay, so between DNA and RNA, this is called transcription. And between RNA and protein, that's called translation. Okay, so those are two terms that you do need to know. So the way that I remember this is like DNA to RNA. There are doctors in the hospitals that have scribes that walk around with them, right? And they write down everything that the doctor says, but it's still in letters, right? So it's A, T, C, G. And then they are just going to write it down. Then your job as a nurse or vet tech or whatever you're going to be is to write it down, and you're going to translate it into words for your patients to understand. Okay, so DNA has A, T, C, G. RNA has A, U, C, G. Okay, so there are no T's in RNA. Okay, so if you ever see a T in any of the letters, it means that it has to be DNA, okay? Or if you see RNA, it means that it has to be, if you see U's, it means it has to be RNA. And then remember, amino acids, when they get stuck together, they make a protein, which is the part of the body that has a job. Okay, so with that, let's look at these questions. All right, so it says, which of the following nitrogenous or nitrogen bases is not a component of DNA? So remember that DNA is A, T, C, G, where RNA is A, U, C, G. So we're looking for which one is not component, and that would be the U, which is uracil. And yes, you do need to know that adenine is A, guanine is G, cytosine is C, thymine is T, and uracil is U. Okay, so you do need to know the full names of those. All right, nucleic acids are polymers of many nucleotides that are held together by what? And this is just something that you should pick up along the way, right? And this is phosphodiester bonds would be the correct answer, okay? And we talked about it in the lecture slides. A DNA helix is held together by what? And again, these are just going to be the hydrogen bonds between the bases. So you're going to have that backbone that's going to be, making that double helix, and the two sides of them together that makes that staircase looking thing. And those are going to be held together by hydrogen bonding between those bases. Again, we talk about that a lot in the lecture videos. Okay. Which of the following is not a major type of RNA? Well, we use messenger RNA, ribosomal RNA, and transfer RNA, but transcription RNA, that is one that is not a major type of RNA, okay? And then it says, what is the sequence of amino acids that is going to be coded by these amino acids? So GUC, AGC, CCA, and mRNA. All right, let's see if I am smart and have give me 1 second, guys. You should have a protein table, but of course I don't have. So pause for 1 second. Okay, so how we read this table, okay? If we have the first letter here, then the second letter, then the third letter, okay? So say we have AUG. The first letter would be A, then we look for the second letter, which happens to be here, and then where they intersect, you would look for G. So AUG happens to be the start codon, okay? What if we have CAG? I'd have C's over here, then I'd look for A, and then I'd see where they intersect. CAG is glycine, okay? GGG, we'll say GGG is going to be glycine, okay? So that's how you would read those. So it says, what is the sequence of amino acids coded by GUC? So first I'd say G is the first one, and then U is the second, and where they intersect, GUC, is going to be valine, okay? And also make sure that they're all U's because this is mRNA. So mRNA should only have U's in it, no T's, okay? AGC, well, I have A here, and then the second letter is here where they intersect. AGC is going to be serine. And then CCA is going to be proline. So then when I look, I should have val, ser, pro. So D should be the answer for that one, okay? And that's on page 597 of your textbook. All right. All right, 17.6. So for this one, it says, which of the following are causes of DNA mutations except? Okay, so we have point mutations, we have silent mutations, and we have frame shift mutations. And I talked a lot about this in the video, okay? So point mutations are if we have like A, C, G to A, G, G. We're only changing one letter or one point, okay? And that can end up changing a whole entire amino acid, or it might not, right? But the point is that it just changed one letter, okay? So one change. The substitution or silent mutation. So that would be like, if we're looking at this table again, we see how we have a lot of different amino acids that are on this table, and they have different letters that code for them. So a silent mutation would be like if we went GUU to GUC, it still codes for valine, right? So it's like, shh, don't tell anybody because I made a mistake, but it doesn't really matter. So that would be a silent mutation. And then a missense mutation is, oopsie, I made a mistake, okay? And so that would be like if you changed UAC to UAA. You're still just changing one letter, but it changes it from tyrosine into a stop codon, okay? So that would be like, kids, stop your nonsense, right? And so things like that. So the point mutation is one. Silent mutation is when it changes the actual letter or the codon, right, the amino acid. A frame shift mutation would be like if we have AAA to AT, UC, AGG, and we end up having like an insertion there, frame shift insertion. So now we have AU, and then this A has to move over. So then now that will be AU, I don't remember what I had that, I think C, and then that would have to move over, or actually it would be AAU, and then this C gets moved over, so then be CAG, and then this G would be holding on, okay? So that would be a frame shift mutation with an insertion. And then codon transfer is just where you're transferring a codon, okay? So it says, all of the following are causes of DNA mutations except, and this one would be the codon transfer, because again, these ones are all mutations at the DNA level, the ATCG, this one I put RNA, and so this one is wrong because it should be actually in DNA, but we'll change that to a C so you don't know. But yeah, so anyway, these should all be in DNA. But the point is that you're shifting in one letter. It's called a frame shift mutation, where it's actually shifting that frame. I used to think of this as like photography. If you're taking a picture and like somebody puts in the middle, everyone has to like shift over to the right to make room for it. So that'd be a frame shift mutation, where if somebody steps out of the picture, everyone has to shift to the left to kind of make up that lost space. All right, moving on. It says all of the following, a typical virus contains all of the following except, and they can have DNA or RNA, but they do not have both, okay? So they can have nucleic acid, which would be deoxyribonucleic acid, right? They can have a protein coat as a protective coat, but they cannot have both DNA and RNA. That's why they have to take over the host cell so that they can kind of hijack it and have it make them for it, because the new genes and proteins for it, because they cannot have both DNA and RNA. All right, in the nucleus, genetic information for the synthesis of a protein is copied from a gene in DNA to make mRNA. Okay, so this one is a process called what? Now remember, we said transcription to translation, and that was the first thing that we talked about, right? And so I don't know what I did with my little piece of paper that I was drawing on, because I actually showed it to you, but it is going to be, that one is the transcription, okay? So if we're going from DNA to RNA, it's transcription. RNA to protein is going to be that translation, okay? So remember, that was the first thing that we talked about when we started talking about this. So this one is going to, again, be transcription. All right, what is the anticodon of tRNA for AGC in mRNA? Okay, so this is just basically what matches with what. So you're going from RNA to RNA. So great, so we just have to make sure that we stay in U's, no T's. And the anticodon just means what matches. Okay, so if I have DNA, and then I have RNA, and then we can have, mRNA, etc. So we have coding strands and, transcript, but there's a lot of layers that you don't have to know about this. But basically, if you have DNA, you would have ATC, GCC, ATA. And if you go to RNA, then that would be A matches with T, but remember that in RNA, we don't have T's, so that A would have to match with a U. That T matches with an A, the C with a G, G with a C, C with a G, C with a G, A with a U, T with an A to a U, because we're going from DNA to RNA. So now this is saying RNA to RNA, so we just have to say who matches for RNA. So we have A, G, C, and it's RNA, and then we're still going to RNA. So A matches with U, G to C, C to G, okay? So that one would be then answer B. All right, which of the following nitrogen bases is not a component of RNA? Okay, and remember, we need to know the full complete name. So DNA has A, T, C, G, where RNA has A, U, C, G. So which one is not in DNA? That would be the T, which is thiamine, okay? And then which nucleotide is not in DNA, okay? This one is gonna be one that we talked about in the lecture slides, but I'm sure there's also, there is the table and stuff like that. The one is not GMP. They're the little guys that are like the DGMP, but just straight up GMP is not the correct answer, okay? All right, what is the complementary base segment of DNA for this sequence? So it's saying DNA to DNA, okay? So if we have A to T, C to G, and we write this down, C, T, A, G, G. And usually they're in threes, but they decided to do it in 5. C matches with G. T to A to T, because we're still in DNA. G to C, G to C, okay? So then we should be able to see that. So GAT, CC is gonna be answer C right there, okay? All right, what is the complementary base sequence in mRNA if the following sequence is in DNA? So now we have to remember we're going from DNA to RNA, okay? So we have T, G, A, C, A. So T matches with A, G to C, C to G, C to G, A to normally T, but since we're in RNA, it has to be U, okay? And so that one should end up giving us which answer? It should be ACG. Why did this do that wrong? ACU. So this one is correct, but why did I have a U there? Oh, because that's an A, because my brain decides to be fun. All right. So there you go. Sorry, the original DNA was TGACA. So T to A, G to C, A to U, C to G, A to U. Okay, so sometimes you gotta check your answers because when your brain has fun, your brain has fun. All right, so that wraps up chapter 17. Hopefully that was helpful.